School of Life Sciences, Bhupendra Chaudhary, and today I shall be talking about one of the themes in sustainable world. Because when we say talk about sustainability, we talk about sustainable world, and sustainable world includes many things. Among them, there is one area that we talk about crops, right? Crops means plant, and because the human population is largely dependent upon plant-based foods, right? We have two major sources of foods available. So, and my introduction is this: that I come from School of Life Sciences, and I am a learner, right? That is my introduction. If you want to see more my academic credentials, PhD, postdoc from US and whatsoever, that is there on the website. Just go and check it. Learner means we all are learners here. We need to learn, right? And those who are learning, they are science people, right? That is a very good introduction that we all should have. And I take you all as a learner. Those who are missing here, I give them 1 mark less to be a good learner, right? So they need to improve upon their habit, because if you are a good learner, you go search for knowledge in library, in class, in lectures. हो जाएंगे sign, don't worry, focus here. Okay, so so today I'll be talking about, as I mentioned, that there would be different aspects of sustainability, and among them you have food. The food is there, the world is sustainable, right? And these days, you have climate change, global warming, other CO2 release. So though there are many factors, those are affecting the climate. So that means our crops that we are using for our food, especially for food and fiber and oil purpose, broadly for food purpose, should be protected from these kind of stresses, isn't it? So that's why we always learn about that. We need to develop or modify our existing crops for modified aspects of making more produce, that we say climate resilient crops. Okay, so I'm just making these key terms in your ears so that you are comfortable with these terms when we talk about it. So you have to have climate resilient crops available on the globe, so that can bring you more food for this hungry world. The world is hungry. सुबह खाया कुछ? Everybody needs it. Every animal species needs food, right? So they search for food, and we people are sitting here in AC room without worrying. If you go outside this hall, you are having some birds, a number of species. They all already have started their search of food. We haven't. Why? एक चिड़िया सुबह उठ के खाना ढूंढने जाती है, वो दिन भर ढूंढती है. आप देखिए, the movement is always going out, coming back to nest, feeding the young ones if they are there, or eat. हम तो नहीं करते. क्या रीजन है? So this morning we had भाई टाइम से क्यों नहीं आते? अंदर आओ. भाई रिकॉर्डिंग जा रहा हो. How many of you had breakfast this morning? Those who came late. Breakfast किया? क्या खाई? Bread. So you have some calories in you, right? क्या खाया? Bread and omelette, and someone may have aloo ka paratha and whatsoever. So this is food, right? And we had it. And you go to lunch. You have lunch here in SBT canteen. मिलेगा? कैसे पता? कल मिला था, छोड़ दो. आज मिलेगा? बताओ. मिलेगा? How do you know? Did I tell you? No. The teachers told you? No. क्यों मिलेगा? आप कैंटीन जाते हैं and there is nothing on the bench. What would you do? What is the guarantee that you have food available on your plate next meal? Lunch, dinner, maybe tomorrow morning. How do you know? A bird doesn't know. It is flying all the time to nature and collecting food for breakfast, and then started for lunch, and then started for dinner, and then sleep. And next morning, get up, again search for बारिश हो गई तो नहीं मिलेगा. It has to wait till lunchtime. Right? How do you know that you have food in canteen in lunch? That means there is a difference. Right? So we know that milega. That means we have some security in our head that food is plenty and we have already produced enough food and that I have reserved in my bag and I'll be getting. That means there is a food security available with you right now when you are sitting here talking. Right? Security hai? Ghar jayenge to khana milega. That is security. If you are secured with something, if you have 10 rupees in your pocket, you are secured that I can go to canteen and buy a tea. That is a security. Right? That means our brain, we are sustainable people, sustainable system, that food is available and the security, the food security is there. कहाँ से आई? If I see a data right now, the food security that we are talking about right now in 2026, this is an estimated, a projected data that if we go to 2050, you would be on peak of your career at that time. I'll be retiring, right? Retired, 43. So, so this is a projected data for few crops, and this is true for every crop. In next 25 years, I would say, less than 25 years. So you would be having, and you know, the hungry stomachs are increasing every year, right? That means human population is increasing and everyone needs food, fiber, oil, everything is required by everyone, right? My plate should be decorated with rice, dal, carbohydrate, protein, then some fat, then some salad, everything. And you can imagine how many constituents are required to decorate your plate. I need a decorated plate. Why not? Nutritionally rich, diverse in different food components, and for that you need to have more food because there are more people to eat on this globe in coming time. They are increasing. They are increasing. We say that we are 140 crores, maybe 150, 160, and the global population is increasing as well, right? So this is a projected data that maybe in coming time, coming few decades, 2 to 3 decades, we would be needing a drastic jump in the production. If we need to keep that security in our head, that food is there in the canteen in lunchtime, right? So if you need to maintain that security, you need to increase the production. And the projected estimation is that 50%, 60% in wheat. Can you imagine whatever wheat we have today, we have to increase at least 61% more of the existing produce in coming 2 to 3 decades, right? Corn, sugarcane, chini bahut khate hain, 87%. And now sugarcane has been used in making ethanol, bioethanol. That means the production is needed more. This is for sugar purpose only, because we celebrate a lot and we eat sugar and sweets. So now if I add bioethanol demand in here, it would go more than 100%, because now we have started using E20 and then E30, and now E85 has been announced last 2 days back, and the target is E100. Ethanol 100 means you are having a lot of bioethanol coming from sugarcane. The major source of bioethanol in our country is largely, we don't have corn that much, but sugarcane is the primary source. That means you can imagine, and then there are more crops. You cannot list everybody on the board. So you can imagine that you need to increase the existing production that much, maybe more in coming decades to maintain that food security. Food security in 2050. And if you talk about 2090, and where does the food come from? Come from land, agri-land. And what we are doing? We are using agri-land for some other purposes, because that is required. Sometimes we say highway is being built. Sometimes we say an airport is coming. Huge land. And that land was previously was being used for making your food, producing your food. And if we are using it for some other purpose, land is limited. We don't have enough land that we can go in sea and increase land area. We have limited land. You can use it for agriculture. You can use it for making highways or airports or commercial buildings, right? That means you are shrinking the agricultural land. Because land is limited, we are using it for n number of purposes. That means the agricultural land, the land that is being used for agriculture is being shrinking, is being reduced. If we talk about in area, right? Area under cultivation is going down. At the same time, population is increasing. That means demand is increasing. So production area is decreasing, demand is increasing. That means you have a tremendous pressure on agri system to produce more in a lesser area. Getting what I'm saying here? You need to produce more to bring that food security. Either you say, I don't know for tomorrow, and let's finish our food, and then the famine situation will come. I'll talk about it when it was there. You did take it back. Or if we need to bring food security, we talk about these. We ask these questions that area, and you can say that that is development. So in the name of development, if I'm using land, that is my choice, right? So whatever is being happening, we have to balance it out. Balance it out in the sense that whatever land we have, save it, and then produce more in the limited area. That is a challenge. Challenge on whom? The plant breeders, the plant scientists, the plant biotechnologists, right? So there is a tremendous pressure. We don't even care. We go to our food mess. Maybe you are using it, or at home or at restaurant. You can see how much food is being wasted. We don't realize that it comes from a chain. The person is sowing it, ripening the crop, harvesting it, transported to your plate, and then you are losing it in the plate. And that food is required for tomorrow. Because the scenario that I'm showing you is challenging. Oh, you would be having enough money in your pocket, doesn't matter, but you are not having enough food in the market. What would you do with that money? Because food is not produced, food is not there. Government of India has stopped recently, must have stopped exporting sugar, chini. We are rich in sugarcane cultivation, so we used to export it, and export it you get money, right? So a lot of revenue is generated. So stopped maybe 2 months back, because that first we have to feed our own people, and then we say if we have surplus, then only we go. That means I'm sensitizing you here, right, about the challenges that we are facing right now. And in coming decade, you will be seeing them in the market scenario. As you can see, the gas prices are going high. That is increasing inflation. And of course, the food is becoming scarce in coming time, right? So, and there was a time, don't forget that Lal Bahadur Shastri ji was our Prime Minister in 1964. He requested, at that time we were 44 crore only. He requested everyone to save a meal in a day, that only take 2 meals in a day, save one for tomorrow. That was the situation. And then somebody came, and then we got that food security. I'll be going to talk about this right now. And this is what just an introductory slide, that whatever crop we are using as a source of food or fiber or oil, we are basically they have been evolved, they have been originated at a different part of your globe that we call center of crop origin. And you can see they are diverse. Fertile crescent is the one where we belong to, right? That is very fertile area for crop evolution. Lot of crops evolved from that point and then diversified across the world. And you can see maize, wheat, rice, pea, and there are many. This is only few. So whatever you say, Mexico, Peru, Fertile Crescent, China, these are the centers of origin, and then they diversified. Whatever cotton we are using right now, I'll be talking about it, was originated in Mexico. The very first time they came into Africa continent, and then they moved. I'll talk about it in the coming slide. And then whatever cotton we have right now, because we are second largest producer of cotton, and we are making a lot of cotton products as well in textile industries, and those are basic. So you have different components, different crops have been evolved under different centers of origin, and that's how they are existing right now. So when you talk about those challenges, as I was talking about, so you need to improve it. Okay, so in the given area, given agri area, you need to produce more, and if that is the case, you have different areas that you need to work upon. And I have highlighted 3 major areas that you need to work upon. The first area is that whenever produce is being made in the field. Let's say we are using wheat, rice, or tomato or barley. So whatever is being produced by the plant, you don't harvest 100%, right? 100% it doesn't reach to your plate. That means some percentage of the produce that plant has made. Plant has done its duty. Plant has made. Suppose you have a wheat plant. Have you seen wheat plant? How many of you seen? किसने टच किया? When it is in field and it is blooming. जब सीड आ रखा हो, टच किया? नहीं किया? किसने किया? आपने किया? टच करके देखा? Right? सबने देखा? नहीं देखा? Right? But we eat every day. You can imagine the scenario. We eat every day, and that is our major staple food, and we haven't even seen it. Cultivation is a far story. We haven't even seen it in the field, right? And we need that food security. Somebody else should do it for me, and then I am secured on my plate while sitting in the AC room, right? That is actual scenario. You can judge ourselves. That I'm part of you, right? So that is the situation. So you need to decrease. So plant has, let's made 100 seeds per plant have been produced. That's an example. Okay. Stop me if you have any concern or question anytime. So whenever your time is done, let me know. My slides, it's a bottomless bucket. It won't stop. Okay. So you just tell me to stop here as per time. So let's say wheat plant has produced 100 seeds per plant. Now, when you harvest it, it's mature. Some seeds fall down. When it is in transportation, some got fungus. When it is being threshed, something fell down on the ground. And you ultimately harvest 80 seeds, let's say, per plant. So 20 seeds plant made, but you got ultimately the production that we say 80 seeds. Whatever I got in my bag, I counted that this is the production, annual production of wheat this year, right? Plant produced more, right? So when we talk about that we have dearth of food, the very first question comes in, whatever production we have, are we harvesting it 100%? You say, no, there are fungi, there are bacteria, there are nematodes, there are insects, there are drought, there are salinity, there is harvesting problem, there is transportation problem. So you have n number of factors, those are influencing your harvest versus production. How much is produced by the plant and how much you have in your bag, you have a difference. And that difference we call losses. We lose production by any means, doesn't matter. Fungus agi, you have vaccinia that is causing rust and astilabud that is causing smut disease on cereals. Huge production goes out due to those stress. That is we call biotic stress. Now we have heat stress. March was hotter like anything. Wheat plant needs little cool temperature, right? It's a rabi crop. Rabi crop means it starts from November to March or April. If temperature goes high, that we call heat stress. Drought, pani hai niche, mitti mein pani hai, but plant is suffering with heat, high temperature, and that affects seed production, and that affects yield production. That means we are losing it due to certain factors. So we can control those factors, we'll talk about it. But the very first part is that you can decrease those losses. How much we can? Market is jate ho aap. Sometimes you go for shopping, you buy tomatoes. And when you are buying tomatoes, do you buy a pulpy tomato that is jo pilpila hai, pulpy? You buy tight tomatoes so that it can last for long. You can come home and then save it for a few days. जो pulpy tomato है, वो तो गल गया. या तो हॉस्टल में आएगा, मैस में आएगा जहां आप रह रहे हो, right? या फिर फेंक दिया जाएगा. That means plant ने तो बनाया था, I could not harvest it, I could not use it as a consumer, but plant produced it. Maybe in transportation, when you have a carton, the lower layer of tomatoes, mangoes, that goes bad because in transportation that become pulpy and they over ripen and then you lose them. Loss hai. That is a loss. Can we save it? Wheat and rice, they have a shattering property. When it is mature, you have a natural process existing that you said dehiscence. So every fruit has to dehisce. Dehisce means it has to liberate seeds out. That is a natural process so that the species can be maintained and the seed can go out in the field for next generation. Fine. But that shattering character, if wheat inflorescence shatters, the seed goes down on the ground. And if the seed goes on the ground, you cannot go back to farm and collect each and every seed with a forceps. Right? You can only harvest it, thresh it, and whatever comes in. Whatever seed fell down on the ground is gone. That means even if it is 2%, 3% of total production, you are losing 3% due to shattering character. Can we improve it? So we can decrease losses. Those are happening there. These kind of traits. Those are happening due to stresses, biotic or abiotic stresses. Those are happening due to transportation or any other issues. And those are happening and we leave food in the plate. So if I collect that food from you 30 people, 30 है नहीं वैसे, 50% भी नहीं है. Where are the others? Why we are running this show? Okay, I'll report to your coordinators that you know climate and then maybe they are in Taj Mahal this morning. Maybe. So do you have an attendance system? Yeah, please circulate it. So I know at the end that who is पहले आगे देते हैं. Circulation माने it goes in a sequence, right? Anyway, आ जाएगा. So decrease the losses. This is my primary concern. If you need to increase production, if you need to bring food security, whatever we are losing, by any means, say it. And the first mean that we can do here is, but when we are sitting in this hall, that don't waste food in your plate. Because there are hungry people, those who haven't have it this morning. That is a loss. Save it. Save it for tomorrow. Okay. So if you decrease your demand, that will help you maintaining the food security for others as well. So let's take a vouch here that you know. Let's vouch for it that we are not going to waste food now onwards. Do you do it? करते होंगे. Rice ले लिया, नहीं खा पाए, छोड़ दिया. Right? That means you are losing the production. We can decrease it. We can bring food security. Okay, and there are different ways that बोल मैं रहा हूँ प्यास आपको लगी है. This is called the consumer's demand, right? That we need everything. उसकी तरफ मत देखो, आप भी तो पी रहे हो, right? मैं भी अपनी बोतल लेके आया हूँ. That this is consumer demand. We need it, and being a consumer, I demand for it. Consumers demand, right? Because I have money in my pocket, give it to me. Whether I eat it or I discard it. No, I'm going to do that because the scenario is very gloomy. I just talked about that. Remember this lecture when you are in 2050 that we talked about it. Even if you do not belong to plant sciences and you are not going to work upon it, doesn't matter if you are a software engineer sitting in an AC hall with a computer and doing some programming for a MNC, but you are going to back home and to have a good dinner with food. And food comes from largely from plant sources, right? So remember it when you have a plate in front of you that you are a consumer and you are demanding for this food. And if you are demanding, someone else is working for bringing that food security. And who is that someone else? It's those people, those who are linked to plant sciences and plant breeding area. I'm one of them, right? I'll be showing some data with you. So first, decrease the loss. And I'll be talking about in detail that how can we decrease the loss with an example in the field, how people have done it. Okay? Take this lecture as a conceptual clarity lecture that, you know, you have these there are challenges and how people are addressing it and what can be done in future, right? Then we say increase yield. Whatever yield we have, let's say 100 seed per plant. I just gave an example. Let's make 120. Okay? So even if I lose 10, 15, 20%, whatever I'm losing, my net production would be higher than what we have now. That is another approach that we are, another parallel approach we should work for both. That is basically increasing the food security chances, right? So you decrease the losses, add the loss to your production. At the same time, you are increasing more. Okay, that is another strategy. And then you say third part, once quantity is enough in your plate, quantity, then you go to quality part, and that we say nutritional aspect of your crop, your food. If your rice is having good vitamin as well, it's a very good source of carbohydrate. But if it can have more protein or more vitamin, that would be fantastic. Or the quality of or the nutritional quality of your food products goes higher. That is amazing. So once we have good quantity, we are secured. We parallelly work upon quality part as well, that we say nutritional improvement. So I'll be talking about all three areas with example that what can be done. So I have produced so far, put forward some challenges, some actual scenario, some challenges, and some broad solutions to you that what can be done. Okay. So are you agree that we need to decrease the losses by any means? There are some losses that I cannot control, but whatever I can control here, I do it. At the same time, through using genomics, biotechnology, and breeding exercises, I'll be talking about increased yield. So the first question is that how can we save the losses? Okay, because there are three aspects. So first part is that I'm going to focus upon the saving the losses. Okay, so this is a basic picture. I'm not really sure that how many of you come from a botany background. We have. Okay, few students are there, and some are from other background, right? Geology or microbiology, something, right? Biotechnology. किससे आते हैं आप? Geography से. Marine biology. Okay. Marine biology. Okay. So perfect. So this is a picture that is showing you how plants have been converted into crop plants. God made plants, we made them crop plants. If you see wheat, whatever wheat we have in the field is not workshop में हो कैसे आना हुआ भाई नहीं थोड़ा लेट हो घूम के आओ कैंपस इज बिग जाइए So how plants have been converted? If you see wheat, I was saying, and wheat was made in nature. Whatever wheat we have with farmer is not the wheat that was made in nature. We made it. Whatever wheat was made by nature is called wild wheat. Whatever is plant is there on this globe, we call them wild plants. Okay, so we have wild plants of everything: wheat, rice, maize, cotton, everything. We have wild plants. And then we modified them for what? For the produce that we are looking for. In wheat, what do we see? We see seed, grain. So number of seeds, if you see in wild wheat, the very first picture. I don't know if you have cursor here. Here, this is wild wheat. This is modern wheat. And if you see number of seeds wild, you have, because each plant is producing few seeds. Few money 5 to 10 seeds. If one individual in nature is making 10 individual next time, the species would never die. Right? That is the species, and every species would like to survive on this globe. Those who are from diverse background, they would be happy to learn these kind of statements. So, if one individual is making 10 is making 100 next time, that means species will survive. And that is sufficient to survive in this nature. What we did, we said, oh, if I just crush this seed, I get some powder, white powder. And if I eat that white powder, that gives me some energy. Okay, so those 10 seeds were going to field to nature to make 10 plants. I didn't allow that. I crushed it. So seeds are not going to nature. How a species would survive? That means to get me food, I need more seeds, so that some seeds can go to nature next time, that we say next generation, and some and maximum of it I can crush and make flour and make chapatis. Okay, so for that the essential part is that I need to increase number of seeds. So what has been done by people, by human beings for last 10 to 20,000 years, that they had started acclimatizing or adapting each crop, each plant for the specific produce. Wheat for grains, rice for grains, cotton for fiber, mustard for oil amount and oil quality. Quality comes later, oil, right? So you have different crops. Maize for seed, corn. You can see the picture in here. The first picture is wild corn. This is modern corn. So you can see that that is a very good food in Western countries. That means we have started modifying the existing plants as per our need to improve something upon the specific produces. Okay, and that process went upon 10 to 20,000 years. And how did we do it? We means the people, the human beings. 10,000 years back, people have started selecting plants and modifying them. And that process was called domestication. So we had domesticated animal species as well as plant species with us, because we were also evolving with our practices, with our civilization. So suppose you have 100 plants of this kind wild, so you would be having a natural variability in population. Do you understand what natural variability is? If we have these 15 students sitting here, and I go by eye shape, ear shape, skin color, height, you take any character, you have a variability. Lowest to highest, left to right, right? That means for any particular trait, you have a variability in nature. You can see anything, you take any trait and you have a variability available in among individuals. So we use that potential of having that variability in wild population, selected the best one. Suppose you have 100 plants and they are producing number of seeds, let's say 5, 6, 7, 8, 9, 10. This is the range. Some plant make 5 also, some plant make 10 also, and there is a range. So what I did, I selected the best one. I took the 9 and 10 one and allowed them to go to next generation. Others were not allowed. So we grew the best plants to next generation. So again, I got 20, 30, 50 seeds, grew them. So the plant that was having 10 seeds per plant in previous generation would go at 11, 12, 9, 8 also. So now range has gone from 8 to 12. It was 5 to 10 earlier, 8 to 12. Then 10 to 15, 15 to 20. So if you do it for next 20,000 years, what would you come from? You have modified a plant, selected a plant over generations for a particular produce. And this is called adaptation to the selection. So it's a human selection. There is something called natural selection that is happening in nature. There is something called we selected. We do it in breeding. This is basically classical breeding aspect. We have modified it now with more information. So this is the process. Those are from different background, not from plant. That is called crop domestication. And what has happened in that? How number of seeds increase? Because we know that everything has a genetics. You have genes available for every trait. So what has happened? So the gene pool concept says, this is a gene pool concept shown on the board, where the darkest circle at the center is basically those are currently present in the field right now. Whatever crop we have, every crop, where we say modern crop species, domesticates, जो आज domesticated हो चुके हैं, those are in the field anywhere in the world, doesn't matter. So those plants are basically constituting the primary gene pool. Gene pool माने all the genes, and every gene has alleles. If you know, different copies of those are from different background. Different copies of different genes are called alleles. Okay, so you have different alleles. Let's say genes, just to minimize confusion. So whatever genes are currently present under cultivation. For example, wheat. We have some wheat growing in India, North India, South India, East India. Something in Japan, something is in Ukraine, something is in USA, something is in Europe. So you have all the wheats growing right now constitute primary gene pool. So all the genes present under cultivation constitute primary. And why do we call it a gene pool? Because we can shuffle those genes from here and there. I can take a Japanese cultivar of wheat and breed it with my Indian cultivar for a better resistance against stresses or better production. That means we can move genes from here. So that constitute a gene pool. That we say primary gene pool. Under cultivation, those are still growing primary gene pool. Now you have some crop forms, I would say, those were left behind, right? So if you go the middle circle, there are more cultigens. Those were kind of parents of modern domesticates. In the process of 10,000 years, so there were some primitive forms. Those were intermediate between the really wild one and the domesticate one. That we say early cultigens or land races. There. So they are not under cultivation, but they are still there in some repositories or some laboratories or some part of the globes if they are endemic to certain area. So we have some more genes in there. So we can bring those genes also together if required sometimes. Suppose you are looking for resistance against Yersinia to make your wheat rust resistant. So you can bring within primary gene pool. Sometimes you can bring from secondary gene pool also. And the wild species, those are there in nature. Still they are in nature. constitute tertiary gene pool. So there could be many other genes, those are still there, but not part of it. This is a gene pool concept for any crop. So here, as per need, I can move genes and characters from here and there. Suppose my wheat is sensitive to rust disease. Okay, so I can look for some genes, those are available, that can give me rust resistance, maybe from primary gene pool. If available, fantastic. If not available, I can go to secondary gene pool. If not available, I can even go to tertiary gene pool, that is including wilds. So if I have any gene, any allele available anywhere, I can use it for breeding purpose. Okay, that is a source. We can cross them together, go to F1, make the segregating populations, and we can use it. And for that purpose, if you have such a scenario, you use a gene transfer, a character transfer technique that we call introgression. This is the basics of classical breeding. So if you have different species, and that we say, if you have a species B, let's say blue circle, and you have a species A, the red circle, and you say that species A is good to me, the only problem is that it is sensitive to rust, a fungus. When I screen my gene pool, the primary one first, because it is easy to cross, and as you go far, it becomes difficult. So when you have this primary gene pool, and I look for that there is a species B available somewhere that is rust resistant. So what I can do, I can transfer rust resistance character from B to A. Okay, A is good to me, so I can do that in the field, and that we call classical breeding. And this approach, I'm not going in detail. This is a backcross breeding plan. BC stands for backcross. So backcross breeding hota. Backcross is where the hybrid is crossed with one of the parents. That is called backcross. So you make a hybrid, red and blue, half red. That is called hybrid, right? And that hybrid can be crossed with either of the parents. Suppose here, in case of this example, it is crossed with red. So the next population would be a backcross population. So F1 goes to backcross 1. Then you go to backcross 2, BC2 to BC6. And if you do that, there are more details in here that I am not going, because the audience is diverse. And so you can always transfer the trait, suppose rust resistance, from species B to species. This is possible under field conditions. It will take time. You go to up to BC6, means there are 6 to 7 generations that you have to live with. So if you take 2 generations in a year, 3 to 4 years plan, right? So, but it is doable. You can do that. And that process is called introgression. Likha hai. So if you transfer a trait, character, let's say rust resistance is a character. Actually, it is a phenotype. When I teach genetics class, I always define trait, character, and phenotype differences. But here, for the broader audience, I say that a character has been transferred from, that is a rust resistance, from species B to species A through a process called introgression. Classical breeding ka typical example. Okay, this is how you can always do that. And this is how crops are being modified. Okay, do you know this guy? Norman Borlaug. नीचे नाम भी लिखा है. Who was he? जी, हाँ. If we are sitting here and I'm not worried that I'll be getting my lunch in Pata canteen or not, I'll get it. If I had that security, this is the man behind it, right? Who started modifying your cereals, rice and wheat, for food security purpose through classical breeding. इसलिए मैंने आपको पहले breeding समझा दी, that how breeding is done. So using introgression, he has modified wheat and rice crops. कैसे modify करी, and that has actually brought security. So he said at that time, it's 1950 that he made this statement. There are 6.6 billion. Now we are around 7.4 billion on the globe, and we are estimating that we would be 9.2 in 2050. 9.2 billion. It's a world population. 1950 it was 6.6. This is an old statement. Currently it is 7.4. It would be 9.2. जिस स्पीड से बढ़ रही है, it would be 9.2 in 2050. And you have to feed 9.2 billion people. Everyone would be hungry. So he said 6.6 billion people on the planet today. With organic farming, we could only feed our billions of them, which 2 billion would volunteer to die, because we cannot feed everyone. That was the scenario. And they modified it and brought food security. So, and for which he was basically awarded with Nobel Prize for Peace. He modified wheat and rice crops. Why peace, Nobel Prize? Anji? It would have been for agriculture, it would have been for a technology, it would have been for science, something. Yes, if you are hungry, you will fight. If you are satisfied with hunger, you will not fight. Peace will come. Okay, so that will be a famine situation. People will fight with each other if there is no food. That is why he was given Nobel Prize for Peace. Nobel Prize 1970 for peace, and it was the more than any other single person of his age helped provide bread for the hungry world. 1950, and he said that we are food sufficient for next 50 years. 1950, he said 50 years. No one would die with hunger. It's already 76 years now. Okay, we are still running with those wheat and rice that he developed. Okay, but now certainly we need more. And this was the wheat that he has worked upon. And just for your information, the wheat used to be tall, 6 feet, like my height tall wheat. And when it is mature, the crop is mature, it has heavy weight at the top in terms of fluorescence. It used to fall down on the ground. And once it is fell down on the ground, you are not able to harvest it. So a lot of production was being lost due to the plant height. So what he has done, he has modified a tall plant to a short plant by breeding. And now we know that there were RST1 and RST2 genes. Those were basically targeted. Their genetic modification, not genetic modification, their modified version of those genes, sometimes we call them mutated one, is being bred into tall varieties, because we had some short varieties, and the intermediate one, those are of 2 feet height. Whatever we see now in the field is the semi-dwarf wheat developed, and that you could escape from the lodging option. They used to lodge on the ground due to plant height. So plant height was shorter. We never touched upon the yield part. We could only save the losses. Can you imagine? Saving the loss brought you food security for next 50 years. So the plant height was reduced under Green Revolution, and some disease resistance was also incorporated. And the semi-dwarf wheat, whatever we see these days in the field of this height, my knee height, is basically a semi-dwarf. Because we have a dwarf, we have a tall, the semi-dwarf wheat was developed through breeding, as I shown the introgression scheme, and that semi-dwarf wheat is feeding us now, right now. This is for rice. You have rice that is from pita rice, that is basically tall, and then DGW gene, that is from Taiwan, short, and then the IR8, that is a semi-dwarf rice that was through breeding process. Okay, so this is how plant height of wheat and rice was reduced to half of the level, and we didn't take it. 1950 it was made. 1964 we were hungry, and we were not accepting these back cross breeded wheat, because it was called in India, it was called Bona Gehu, and it was semi-dwarf wheat that they developed. When it came to India, it was labeled as Bona Gehu. So people had a notion that if we eat it, we will be Bona. So said, no, I'm told, how can my height can go? Oh, I'm fine. My next generation would be dwarf, because and we didn't accept it. Public hai, nahi sunte, right? So do you agree that if we eat dwarf wheat, our next generation would be dwarf? Right? Do you agree with that? Why? Because we know science now. We know that plant height character doesn't affect human height. Right? We know that it's a food. We crushed it, and then it's only carbohydrate. But at that time, 1960s, 63, we had famine situation in India. We didn't have food to eat. As I mentioned, Shastriji requested everything. So, and then people came, MS Swaminathan Foundation, and then we brought, and we didn't have food. Then the statement was made, if we eat it, we will survive. Whether tall or dwarf doesn't matter, we will survive. If we don't eat it, we won't even survive. Next generation, forget it, that it would be tall or dwarf. Then it was accepted, and we are growing it. So right now. So this is Green Revolution. What we are talking about here is green revolution, and I have already talked about the science behind it. When we are talking about the process of gene transfer, character transfer through introgression, the scheme has been shown. That scheme was taken: a tall plant and dwarf plant crossed, back cross, sixth generation, and then the product was here. Green revolution. So if those coming from marine biology, they should also know about green revolution, right? And what was done under that scheme, right? So plant height was modified to produce more. So we were saving the losses. Didn't it? We talked about saving the losses. What example available? Now we ask another question. That if we have such technologies available and we have challenges available, and I said that in next 20 years or 30 years you need more production, that you can ask that means we need another green revolution, because that drastic change in production has to be there. Answer is yes, we need green revolution in any of the crops, right from cereals, millets, mustard, wheat, rice, cotton, everything, right? You name it and you need more production. So and then I come back to this. If yes, then we already talked about the introgression scheme. The picture remains same. Another aspect that I'm adding here is genetic engineering. So to fasten the process, because it takes time to fasten the process, we have certain genetic engineering approaches available these days. Those are this is just a typical picture where you can manipulate, and we know that DNA genes are responsible. At least genes everyone knows, right? So genes are responsible for every trait. If you need to have resistance against rust or increase production, so there are genes for rust resistance, there are genes for plant height, there are genes for number of seeds. So we can manipulate those genes precisely, and that can give you enhanced production, right? So I'm adding another aspect here along with classical breeding that we used for green revolution, introgression scheme, the genetic engineering aspect. Okay. So this is a basic process of genetic engineering. How do we do it? You have certain genes. The first thing, I don't have pointer here, but you can see the double strands. Those are basically genes or DNA stretches that you can use. You can clone them into some vehicle that we say vectors, and then put it into some other cell like a bacteria, agrobacterium or something, and then you transfer it to the cells, and those cells in the plate, there are cells growing, and those cells can be converted into plantlet. That means the process tells you that you can put or delete, insertion or deletion, a particular DNA sequence from plant system as per your objective, as per your interest. If suppose you have a gene available that can give you resistance against rust fungi, and you need to put that gene into plant, you can do that. Suppose you have a repressor, you have a gene that is stopping plant to be resistant. There are certain factors, you can stop it, you can silence it, right? So you have genetic engineering approaches available, and this is a basic picture that you can take gene, clone it, and then use that clone part to transfer the gene into plant system. I'm not going in depth with typical words, because of that everyone can understand, right? So everyone can take this information that this can happen. And to do that, I think the best part is that you need to know that those cells are shown here in this corner can be converted into plantlet. So you should know that how to convert a single cell into a plant system. If you take cell from here, can you make a whole system? You can. This is what is possible in plants. We are still struggling with animal system. Fine. But theoretically, every cell has a potential of converting itself into a plant system, into a system. Plant or animal doesn't matter, because every cell has all the genes available. The genome is all there. All the genes are there in a single cell. Just the only thing that you need to activate those genes and bring the desired phenotypes or desired organ formation and do that. And this process is called regeneration, conversion of a single cell into a plant cell. चिपकली की पूंछ कट जाती है, फिर निकल आती है, regeneration कहते हैं. What is regeneration? That you make more cells and that more cell can make. Our liver system, our liver has a potential of growing. You can take a piece of it, and that piece can make full liver. Regeneration. So a cell can convert into an organ, and organ can make a system. Okay. Animal system we have limitations. In plant system we have very beautiful system available where a single cell can convert itself into a plant. So we should know it first before we put a gene. So what you can do, you can have a cell, and that cell can be supplemented with a gene from outside, and now that cell is called transformed cell because it has taken gene from outside, and that transformed cell now can be converted into a plantlet. So there are two steps in here. First, you put a gene from outside. The process is called transformation, and once the cell has received DNA from outside, it is called a transformed cell. So next step would be conversion of a transformed cell into a plantlet. This is called regeneration. So for each of these two parts, we have techniques available that how to transfer a gene from outside to a cell. There is a system available. I'll talk about it, and then convert a plant cell now having extra gene into a complete plant. That is called regeneration. So ये कहता है जो statement लिखा हुआ है that before you can get a transgenic plant out, you should have both these areas well defined. How to transfer a gene and then how to convert that cell into a plant. दोनों process transformation, the first part. The second part is regeneration. Transformation and regeneration should be available with you. Okay? How's that? Once you have these two things in your hands, you know how to do it. You can always do that. And if you can do that, you can address those questions that we had made. That how to save losses, how to increase yield, how to nutritionally enhance your crop. Right? Those questions. So you can use these techniques to answer those questions. Oh, it moves. So, well, I am skipping this part because of time. So this is like culturing your cells from seed, from tissues, part, nutrient media, and distal conditions are required. So history, I just remove it. These are the two people. If you say how to regenerate a plant, conversion of a plant, single cell to a plantlet, you need an artificial food जिसके ऊपर आप cell को रख सके. And that food was proposed by these two people that we say Rashik and Scoop, MS medium. Medium is an artificial combination of certain salts, and that could be a food for plant cell. In 1962, they proposed a combination that if you add this calcium chloride, ammonium nitrate, blah blah blah, there are 10 components, and then that will be a good combination of these salts will serve as a food, superior food for the plant cell. So if you put plant cells, they can be converting into plantlets. So that's why these two people, based on their name, MS, the medium is named as MS medium, and widely used medium for n number of crops so far. Okay, so before you convert a plant cell into, now I am teaching you that you need to genetically modify a plant cell under genetic engineering, right? What Norman Borlaug did, that was classical breeding. Now we are learning that we are modifying a cell first, and then we are converting that modified cell into plant cell. So these are the basic potentials of totipotency that one cell can have. All the organs can, the whole system can be made totipotent cells. De-differentiation. If you take a leaf cell, leaf is a differentiated, a modified organ. Leaf is already made. So if you take leaf, so there are cells in there, and those cells are defined to do function like photosynthesis, right? Guard cell to just close it. So you have different defined cells. Epidermis is there, hypodermis is there, spongy parenchyma, palisade parenchyma. So you have those defined cells. Now I am asking those cells in culture that you make another plant. Leaf is not supposed to make another plant, is it? Have you ever seen that leaf is making another plant? Unless you have an auxiliary bud in there at the edge. That I am not talking about. The leaf as such, जो आप बाहर plant देख रहे हैं, right? That is not designed by nature to make another plant. But my theory says that every cell has a potential. So what I do, I take the leaf piece, grow it in the lab, and because the leaf is already differentiated in nature from seed, it is differentiated tissue. So I de-differentiate it. That I bring it back to cell level. Leaf is modified set of cells. Tissue है उसके अंदर, right? So I bring it back to the native state. So I de-differentiate it. And once that we say, callus formation, or under phytohormone we can make cells. And then those cells can again ask to make a full plant, make root and leaf and so on. That is called redifferentiation, right? Competency. So we basically we have to play around these steps by the treatment of different combinations of phytohormones. But plant cells can be treated with that. Anyway, I'm skipping this part because this will take long. And this is culture media that MS has proposed: macro elements, vitamins, sugar, and some support system. Some phytohormones are there: auxin, cytokinin. If you are not from this field, you can even take these names just handy. Say auxin, cytokinin, gibberellic acid, abscisic acid, ethylene. There are different phytohormones available. Those are basically used for plant tissue culture. Well, and by this picture just shows by making different combinations of these hormones, these phytohormones, you can decide the fate of your experiment. If you need roots, you can manipulate. This is auxins, Na, BA is benzyl aminopurine, that is cytokinin. So by combination of these, you can always decide that which hormone to increase, which hormone to decrease. So combinations, you can decide you need roots, you need shoots, you need callus. Callus is like this mass at the base. So you can see that what do you want. Accordingly, you can manipulate, and the expected outcome would be there, what you are actually looking for. Okay, so this is just a picture showing you different phytohormone here treatment. Under the first section only, where you are talking about saving the losses. Another beautiful example that comes in cotton. So cotton sabne dekhiye. This is a fruit called ball, and that is opened ball that we have fibers. These are dry dead tissue that we harvest and we use to make thread, and thread is used to make the fabric, right? So another very good example in literature where people have so cotton is basically cotton shaped human civilization. We are the only species that is using cloths, and cloths come from cotton. Cotton is considered as the root driving force for human civilization, right? So here the problem is, so as I was saying in the beginning, that the area under cultivation versus production, that is important. How much area we have and how much we produce. So if you see these some statistics here, picture ke upar. So if you have China, India, US. There are three leading countries in the world, those who are producing cotton worldwide. And if you see, that is numbers are in 1,000 metric ton. 1,000 metric ton. So you have almost 6,000 metric ton cotton production in China. 23 ka data hai, thoda purana hai, but it takes some time before it comes into public. So you have 6,000 metric ton production in China. We had 5,000. What is the come? China, in US has 3,000, right? So we are good. 6,000, 5,000. We are number 2 in the world. The alarming situation is when we see the area that is being used for cotton cultivation. In China, you have 3.1 million hectare land used for the production of 6,000 metric ton. 3.1 million hectare. What we had used? 12.9, almost 13, almost fourfold. 4 गुना लैंड हमने यूज करी, प्रोडक्शन फिर भी 1,000 मीट्रिक टन कम ही रहा. If I compare area under cultivation versus production, where should we have been? 4 times of this. If we have 6,000, we should be 24,000, because 3 goes to 12. So more land under cultivation. That means we are struggling, right? We have land. It's not that we don't have land, but we are having less production. If we had that statistic, but I just projected, we would be number 1 in the world. And number 1 means leading. I mean, no one can even chase it. Like Milkha Singh, running away from everyone. But we don't have that situation. Where are we struggling? We are still number 2, not a bad situation, but we could be number 1 easily and with a leap, isn't it? That is it. And this problem was addressed certain to certain. And why is that problem? Then we say that what are the factors available? And on cotton, in India, worldwide, we have a lot of insect problem. So insects are affecting those who are from farmer community might know that cotton is largely affected by insects, and they eat your fields, not a plant. They eat your fields, and that's how in India especially insects are major problem. In US also, they are also struggling. So and insects are affecting the yield. That means we are losing. Plant is making, but so that is a loss due to abiotic stress. So what was done by an American company? आपने नाम सुना होगा, Monsanto. नाम सुना है? Monsanto. किसने नहीं सुना? Raise your hand. ईमानदारी से, कोई बात नहीं. If you are from marine, I expect that you don't know about Monsanto. नहीं सुना? So Monsanto is a big company. Big money. It's a big shark in the field. Shark इसलिए कहा क्योंकि दूसरों को खाते रहते हैं. They eat small companies. Monsanto is a big company of US, and they are into plant business, crop plant business. They make new products in that and then sell it to US market, sell it to world market, including India. So what they had done, this paper came into 1991. Frederick Perlek, कहीं नाम लिखा है? Yeah, the first author, Frederick Perlek, was the director of Plant Transformation Wing. I met him once when I was in US. So they developed a cotton plant that was resistant to insects. Now, so the scenario is that you have a cotton plant, insect come and eat the leaves, eat the fruit, and that's how fiber is being affected. So, what we do, we spray a lot of chemical pesticides, and a lot of chemical pesticides go into soil. They contaminate your ecosystem. All the insects are not affected by. So you have problem in the field. Okay. So they address that problem, that don't spray chemical pesticides. They are not nature friendly. What you do, you modify the plant genetically. That can save your plants from insects. And that's how that's a huge event, huge product made by Monsanto in the world. And we are also having that. So what have they done? And if you see that if that biotic stress is being managed, what are they doing? They are minimizing the losses due to insects. So they are just simply allowing plant to make more cotton. That's it. So we are stopping the stresses. We are stopping the losses that is happening due to insects. As we had an example of fungus in wheat, right? So and that's how they developed a cotton that was named BT cotton. नाम सुना? हाँ. So BT cotton Monsanto का ही product है. Okay. So if you have heard BT cotton, that means you know who made it? Monsanto made it. Okay. So BT cotton is basically a genetically modified cotton, but a layman where a gene from a bacteria has been transferred into cotton plant, and the bacterial name is Bacillus thuringiensis. BT comes from genus का पहला letter and species का पहला letter. Even if you forget bacteria का नाम, doesn't matter. So BT stands for a bacteria. वहां से एक जीन निकाला जिनका नाम था Cry1AC. Okay, CRY, that stands for crystal. And 1AC, no, so Cry1AC gene is taken from BT and then transfer into cotton. As I mentioned before, that you can transfer a gene from outside to a plant cell, and once it is transferred, that cell can be converted into a plant cell. That was done by Monsanto. Bacteria से gene लिया, cotton की cell में डाला, and then that cotton cell was converted into plant, and that plant was released into field that was labeled as BT cotton, and that plant was giving you resistance against a particular insect called Helicoverpa armigera. लिख सकते हो ये नाम, याद रहेगा आपके लिए. This is how it was made. The leaf piece was taken, it was treated with gene, and that gene was basically before that was cloned into a vector, and vector into a bacteria, and then it was transferred into plant, and plant was converted in the lab into somatic embryos, and through embryogenesis that was converted into a plantlet. So this is the story of making BT cotton, and this is how it works. So you have a basic thing. You have a bacterial cell. This is a bacterial cell. oval one. The square one is basically a plant cell. So there is a system available in plant sciences where you have a bacteria named Agrobacterium tumefaciens. If you transfer a gene into that bacteria, your desired gene, bacteria is having very good system. It can transfer that gene into plant cell. So this picture shows the interaction between a bacterial cell and a plant cell, and there is a passage in here. You can see there is a passage. So bacteria has that ability, that potential to transfer a gene from bacterial cell to plant cell naturally. What we have done, we have taken a gene from one bacteria, another bacteria, Bacillus, transferred into this bacterium, Agrobacterium, and now this will transfer it to plant cell. It is a past potential. Okay, so this is a cotton cell. So the desired gene that you have from Bacillus has now, through Agrobacterium, it has reached to cotton plant cell. And then we took this cotton plant cell and we underwent the process of regeneration, as I talked about, that where your cell can be converted into a plantlet, and that plantlet was made, and that was called transgenic BT cotton. In the picture, what is being shown on the left hand, you have BT field, the transgenic plants. Next farmer field is non-BT, that hasn't grown BT cotton. So all insects have moved and devastated its crop. There the BT field. This is a real picture. BT field was saved because insects could not damage. Can you see how much loss we could save by making one modification? So these plants were also supposed to make product, that was cotton, but due to an insect infestation, they were losing it. So it was a loss, and we could save that loss by genetic engineering. That we can, and that is net production. Very beautiful example in the world. We have in India, we have also purchased these BT cotton lines from Monsanto by a company in Maharashtra called Mahiko. So Mahiko has purchased, it's a company, purchased in whatever price, God knows, or Mahiko knows, from some seeds from Monsanto, and those seeds were used to grow plant and then breeding to Indian varieties, because we cannot use, this is an American cultivar. It was breeded into Indian varieties, and whatever cotton we are growing these days in southern part of India largely is transgenic BT cotton. That is introgressed from Monsanto's BT cotton. Okay, so that is the only product in India. If someone asks you, that is the only transgenic product in India that is currently being grown in the field. In India, we have only one product, only one transgenic product that is being grown, and that is BT cotton. Otherwise, we are not, our government has not allowed for decades the approval of any other genetically modified crop except BT cotton. Okay. Whereas in US, Europe may become a US, Australia, China. We have many other crops like soybean, maize, rice, wheat, papaya. They have already been approved for their field. Okay. In India, we have only BT cotton. Pata hona chahiye aapko. So this is a beautiful example where we have saved the losses, right? Quickly, we will browse through what time you discussed, 11. So the second part, so the first part was crop improvement. We talked about the first part was saving the losses. So loss was, one example was green revolution, plant height decrease kiya, so you have more production. Where the other example was BT cotton, where one gene was introduced from Bacillus to cotton, and then that saved a lot of production worldwide. Worldwide, not only in US. Whomsoever had taken that line is basically the second part was that let's increase the annual yields. Okay, so whatever plant is making, an example comes from my lab because I work on cotton. So we have tried to modify the plant architecture, the plant genome, and then increased fiber production. Whatever plant is making is my control. Then let's see if it can make more. Okay, so that is another example that I'll cite here in this. But before we cite, we should understand that what is the system, what is cotton all about. Before we modify something, we should know about it. Okay, so this is what we did. So if you see this picture, this is how cotton system is evolved. Okay, So we should know that, what I'm modifying, where does it come from, and what are the constituents available, and let's if we can modify those constituents. Means genetic constituent I'm talking about. So previously, like sometimes back, the ancestral cotton evolved, and whatever numbers are written there, A, F, E, B, D, K, G, C, they are with different genomes available worldwide, and many of the genomes came into Africa, and then they diversified. So it is believed, because it's million years story. It is believed that A genome from Africa and D from Mexico. D is in Mexico, right? So A and D met together, and the modern cotton was made. It's a hybrid between A genome cotton and D genome cotton. So the first hybrid was formed in Mexico. And now you can ask me another question: how A traveled from Africa to Mexico? Million years back, right? So that's a question. But if you see the picture, million years back, continents were not that far that they are right now. They're closer. So it is believed that some cotton had fallen down in water, and because it is having fibers, and fiber they don't absorb water so easily, and they swim to Mexico. So, but anyway, whatever, or maybe some travelers came. I don't know if someone was there that time. So A and D, because we have genetic evidences now that A and D are together. That means sometimes back they were crossed in nature. So if you see modern cotton, you can see A and D together. It's a hybrid, what we have right now. And then there are different genomes. So A and D basically merge together. The green box is D, the purple box is A. They merge, and then there are more species. They merged together 1 to 2 million years back. And the first product that was made now diversified into 5 different species. Million years back, 1 to 2 million years back. So out of these 5 species, whatever we are having in the field right now in India is Gossypium. Gossypium is genus of cotton, Gossypium. So Gossypium hirsutum. Okay. So out of 5, only Gossypium hirsutum. शॉप पे जाते हो, कहते हो ना 100% कॉटन शर्ट चाहिए, राइट? क्या नाम है? अभिषेक. सो अभिषेक शॉप पे गया और उसने कहा आई नीड ए कॉटन शर्ट 100% बिकॉज उसमें ना स्वेटिंग नहीं होती. मांगते हैं ना, यहीं से आएगी. तो पता होना चाहिए कि सोर्स से आ रही है. सो A and D came together, and then 5 species are made in nature. Out of this, G hirsutum, hirsutum is the one that is being domesticated for longer fiber, along with barbadense. Out of 5 species, only 2, hirsutum and barbadense, were domesticated by human beings and are currently being under cultivation. Out of these, hirsutum is in India. Some depleted species are also there, but hirsutum is basically covering our field that we are talking about, million hectare land previously. And this is how it is evolved. I mean, it's interesting to see at your level when you are exposed to the basic biology of cotton evolution. You had wild forms. You can see it is a shrub. Herb, shrub and trees. It is a shrub. And then near to a house, primitive house, you have some plants growing. Then someone is harvesting it. Then this is the modification. I mean, if you see, this is wild cotton, right? Modified, and this is modern cotton. You can see more flowers. You see hardly you have a flower in here. This is how produce is being modified under domestication. What we have seen there, and car stands there, works as a reference for the plant height, right? It's not that it's a camera effect. So you have modified cotton for enhanced production under domestication, right? For million years back, I mean, not million years back, it was evolved, and then under domestication. This is a domestication story. Wild form to cultigens to land races to modern varieties. This is like past 10 to 20,000 years only that these crops have been modified. So, and this is the change. If you want to see what has changed under cultivation, this is a beautiful picture clicked by me a few years back, where different species are kept together. Center you have a seed. If you know that you have a seed, and from the seed you have seed surface, you have cells elongating that we call fiber, cotton fiber. इससे thread बनता है. So seed के जो epidermis है, that epidermal cells, they extend and make it's a single cell. If you see one fiber, it's a single cell. You can see it with naked eye, right? So you, from the surface, the epidermal layer cells come out in the form of fiber cells. So this is like they combed and then they were like spread. So this is wild AD1. Wild AD1 is hirsutum. So wild species versus cultivated species. You can see it's a live picture that in the same frame that how fiber length has been selected under domestication. This is for diploid level. So under domestication, fiber length was targeted. If we have more length, that means people at that time they thought that if we have more length, we would be having more fiber, and more fiber, more clothes. Imagine cotton has been started using as a source of cloth at that time, right? So so this has, and if we go back at scanning level, we see that how fibers are being formed. So the surface in first picture shows initiation, that the first surface, that is the basically ovule. Ovule is early stage of seed. Ovule will be a seed after some time. So the ovule surface is now scanned with scanning electron microscopy, and then this surface is showing you these small dots. So they are basically protuberances coming out from the ovule surface, and that became, after elongation, that became fiber ultimately. So from the ovule surface, the cells are elongating and making you 5 to 6 centimeter long single cell, and that is fiber. Then we asked a question that when it is happening, and from wild to cultivated, the cells are increasing, and when they are increasing, when they are elongating, after some time wild it stops, cultivated it keeps on going. What is the genetics behind? What are the genes those have been selected under domestication unwittingly at that time, and that could be further used for further breeding programs. So we asked this question and started working that what are the genes in my lab, what are the genes those are basically responsible for this longer phenotypes, longer fiber production. And for that we had performed a gene expression profiling experiment. That time we used to call it microarray experiment. These days we call that RNA sequencing or something, right? So we performed RNA microarray experiment comparing a wild K101 and a cultivated PSP-7 fibers stage at different stages of development. When you say 2 days old, it's called 2 DPA, 10. So different time point, because fiber length is pretty different. So we have harvested those fiber cells at a particular time from 2 different wild and cultivated accessions, and compared the gene expression profiling between the two at the given time. And we try to find out what are those genes those are responsible for fiber elongation in domesticated animals. Good question. So we are trying to find out that what are the genes those are selected, and if those genes are good, I would reuse them for enhanced production. That was the hypothesis. So what was done? So if you just, I mean, it's few years story summarized on this slide, that we compared different species, different accessions, and ultimately, if you ask me one line, that what are the genes those have been selected under domestication by human beings, I would say there are 3 different classes of genes. Cell wall modi, because elongating fiber is a single cell, and plant cell may it's cell wall. Cell wall, you have cellulose along with some proteins, those are important, right? So you are basically going to have cell wall associated genes, those have been identified, because if you have such genes available, you can have more cell wall formed. From achhi baat hai, we have data available for that. And if more cell wall modifying or synthesizing genes are available, the cells can go longer. If you don't have raw material at all, the cell will stop. And that's what is happening in wild species. Compared to hormone signaling genes, if you have hormones, the cell elongation helps, because auxin and cytokines, not cytokines, auxin, zebulic acid and pulmonary signaling pathways, those are basically phytohormones. They help loosening the cell wall so that it can elongate. So help hormone signaling. And then cell, under this process, a lot of reactive oxidant species are being produced in the cell, and that create a toxic atmosphere in the cell. And so what we found, that certain antioxidant genes are upregulated under selection, under domestication in the domesticates compared to wild species. So cell remain toxin free. So if the cell remain toxin free, then only it can elongate happily, right? So there are different, so I can at least list you 3 different classes of genes: cell wall, hormones, and antioxidant genes. Those have been selected, and those have been upregulated under domestication, giving you a longer fiber phenotype. Okay. Then, then we identified that what are in the first class as I have modified. So today in this talk, I just using the cell wall genes only, right? There are other students those who are working, but and in cell wall genes we identified a gene family called profilin gene family. Profilins are basically they are structural proteins. Those are embedded in cellulose. Cellulose क्या है? Cellulose cell wall में होता है plant cell की. Carbohydrate है. Cellulose is carbohydrate. To give strength to that carbohydrate, you need certain proteins that can give you strength, and actin is one of them. Actin bundles are one of them. They work like you have a fluidicity, and then you have actin molecules, and they give you strength to cell. That strength comes when you spin it down in the form of a thread. It gives you strength in that thread. So one protein that is profilin gene family. Profilin genes are basically the genes. Those are basically responsible for actin polymerization in the cell wall, right? So let me know when I need to stop, because if I can see your faces, you are saying that, sir, enough. We want to take a swim, a bath in, or at least a swim in sea now, because you are a marine student, right? Want to go back to sea. Enough cotton, right? So profilin was taken, that is polymerizing, actin polymerizing protein that is upregulated, 1200-fold upregulated. That means it is helping in when cell wall is elongating. For actin polymerization, you need more profilin, right? And that's why profilin has gone up. It is recorded in our study. You can see my name on the top. That one, and there's another study from our lab that time. And then my PhD student Dhananjay Pandey, corner picture, he was courageous enough to functionally characterize this profilin gene family in cotton itself. Cotton is a tough system when you are modifying. So what was done? He has amplified or taken the profilin gene from cotton itself, because that has been selected, and put another copy of that gene into cotton plant, as was done in case of wheat cotton. They have taken a gene from bacteria, put it into cotton. What we have done, we have amplified a gene from cotton itself and put another copy of that gene into cotton, because we had a logic that profilin gene is basically selected under domestication and giving you a good phenotype. So that might be helpful if you overexpress that gene into cotton itself, and that might give you even longer fiber or more fiber, and that would be net increase in production. Sounds good? So what Dhananjay did, this is how seedling, then leaf explant, then somatic embryos, and then embryos are germinated and new plantlets are being formed, right? So this was followed. So first he thought that if this gene is important, if we silence this gene, what would happen? Okay, this is a basic approach in plant biotechnology, that if something is important, you remove it and let's see if the function is going on or not. If I am important in this hall at all, take me out from this hall and see if lecture is going on, right? That will tell my importance at this moment. That is basically one experiment that we have performed, and that we have taken RNAi. RNA interference is the full form where you suppress a gene through RNAi. RNA, just naam sun lo, I'm not going in detail. So RNAi is a process where we can use to silence a gene. Okay, RNA interference. So we can put another RNA in the cell, antisense, and that can go and bind with native RNA, making a double-stranded RNA, and the double-stranded RNA will be degraded by the plant system through dicer formation, and then so profilin was silenced, and it was seen that this is a fruit at last. Control. This is silenced fruit. That means fiber development has been severely affected. That means profilin was taking an important role in fiber development. पहला experiment हमने किया that it is important. हाँ तो हमने सिर्फ gene expression check किया था. So if it is important, its absence should give you the desired phenotype, and that fiber has been affected. Then we got confidence that okay, if it is important, then let's overexpress it now. Whatever is there, base level is there, let's put from outside. पहले हमने उसको कम किया तो ये हो गया, अब बढ़ाते हैं. ओके, ये हमने बढ़ा दिया, चलो वो भी लेते साथ में. हाँ, चलो आधा आधा लेते हैं. तो ये हमने बढ़ा दिया. तो प्लांट के बाद फिर हमने डाला, और जो पहला ग्राफ है वो दिखाता है कंट्रोल में कितना एक्सप्रेशन था. ब्लैक इज increase. So we have put a gene from outside in cotton, whatever the system I have talked about, agrobacterium and things, and then the protein level was tested, this is transcript level, that more transcript has been accumulated. So you have independent plants. So the black one was taken further, and you can see in black one, line number 7, this is control D, and E is line number 7. Number of flowers. I mean, we were looking that fiber length will be increased. Fiber length तो बाद में आती, पहले तुमने फ्लावर ही देखे, फ्लावर से तो फाइबर आएगा. तो फ्लावर number was high per plant. You have like typical picture, you have 3 flowers shown, you have at least 5 to 6 flowers. Number of flower is what? Number of fruit. Number of fruit is what? Fiber. So the first achievement that we have got here is that we could increase the flower number. Flower number proportional to fiber. So we could increase the production by our target was that we were interested to see that how fiber length was affected. So from this fiber, these flowers, then fruit came up, and we can see that control. The first is fruit ball. इसको हम बोलते हैं. The ball is the fruit of cotton. So ball is control, and then we have transiting line 7. They were normal. They look normal. There was no effect on that. When we compared in the ball, when we compared, this is like opened ball, they were similar. This is ovule, early stage, at 2 days old ovule from control. This was 2 days old ovule from transgenic line. So this ovule is basically where you are giving extensions of fibers. So it was more in transgenic at early stage. But when we saw the later stage, when it is mature, the transgenic line looked alike with control plant. Although early initiation was there, but this initiation was compensated at later stage by control plants. So fiber length, if you see control and OX line, they were comparable. It was not that but flower number was more. So the hypothesis that we started with, that flower will be more, fiber length will be more, is not the case, because early stage it was more, but later stage it became comparable to control. But we could certainly get more flowers, and more flower means more production. That means by manipulating a gene that we identified through some sources, some hypothesis, we can always increase the net production that we started with another objective of this. And then we did more essays, silencing line, and we compared them. We did some gene expression analysis in silencing line, that is for publication. But we could increase. We have some seeds here. See, this is control seed. Transgenic overexpression. Seed size was bigger. This is silencing line. Seed was weaker. So we could get more fibers in terms of production. And there were some genes that we tested later on. So message here is that by genetic manipulation with certain genes, you can increase the net production by any means. By increasing number of flowers, we could not. And now one of my PhD student is currently working on gibberellin signaling in fibers. So we are trying to silence or suppress a gene that is basically a repressor of signaling through CRISPR-Cas. So this is how we are basically trying to modify these pathways or fine-tuning them, and that can give you increased production. Okay. Then third aspect that I need to touch upon is nutrition aspect in next 2 minutes. Okay, so we'll be summarizing. So in first part we have learnt about decreasing the losses by green revolution by BT cotton. In second part we have taken example from my lab where we have increased the net production by manipulating whatever genes. Third part is sometimes we do some essays, we do some experiments of increasing the plant production for nutritional quality. And in that series, the very good example is rice. So in rice is full of carbohydrate. So the idea was that every 2/3 population of world eats rice as a staple food. The idea was that if along with rice we can put some nutrition and vitamin A. Vitamin A is good for our eyesightness, right? Night blindness is cured by vitamin A. Night blindness is a major issue in world. So that is can be cleared with vitamin A content. So if our diet is supplemented with vitamin A, that is not the case with poor people, but they eat rice. The idea was that even if they eat rice, vitamin A can be supplemented with rice can be supplemented with vitamin A, and that can give you more vitamins to the poor people. So in that series, people have modified rice plant with vitamin. So rice is what do we eat in rice? Which plant part? Seed. Anyone else? Endosperm. So seed is a big structure. Seed has embryos, seed coat. So we remove everything. It's only white structure. That is endosperm. Endosperm is reserve food. It's food only. So what was done that in endosperm, yeah, this is a pathway for vitamin A production. Beta carotene is called. So this pathway is missing in rice, and that is why rice is not having vitamin A made at all. Whereas green spinach, green vegetable, they have it because this pathway is working in them, right? So vitamin A. So they saag khaiye. Green vegetables, they have the vitamin A. So that is basically having this pathway. Rice is not having. So what was done? Few genes were cloned. The red one are cloned into rice from outside, and vitamin A biosynthetic pathway was engineered so that beta carotene can be made. That was the effort. And in that process, there are genes. Those are basically, and this was developed. So after the first one is normal rice, and the second one is rice having extra beta carotene, and beta carotene is golden in color. So the rice became colored. That's why it is called golden rice. Golden means the color is golden, and that is rich in beta carotene. And this rice was developed as a source of vitamin. So if you eat this rice, carbohydrate is there along with vitamin. There you are not increasing quantity here. You are increasing the quality here by increasing vitamin A production. Beautiful experiment. I mean, this was the first time where the rice crop was supplemented with multiple genes. A new biosynthetic pathway has been engineered, and the product was tremendous. The idea was another idea. I mean, you can see that what ideas are this is from JNU only. So idea was that potato, आलू तो सब खाते हैं, हर सब्जी में खाते हैं, but आलू is largely carbohydrate. So if we can get some protein along with it, okay. So the potato was supplemented with certain proteins, and protein comes from a weed called amaranthus, right? जो आपका ये नहीं जंगली चौलाई amaranthus होता है, उसमें से एक जीन निकाला AMA1. It stands for AMA amaranthus 1 albumin gene. So that albumin gene will enhance some protein level. So when we are eating in potato, carbohydrate along with some protein, so diet will be enriched. So it was another example where mustard, that is basically source of oil, the oil quality has been improved. Okay, the oil is not good for a heart. So it has been thought of that, if you can modify the fraction, there are different fractions of oil present in it, right? So you have high oleic and जो आप समोसा नहीं आप खाते आता हुआ इसमें कैंटीन में, they are having high linolenic level, harmful for our heart. कहते हैं ना कि कम खाओ, because oleic acid is low. So whatever good oil, vegetable oil you have in the market is having high oleic. If you go in the market and you are having a label on it, just go and check it. Oleic level कितना है, linoleic और linolenic कितना है, right? So you have 18:1, 18:2, 18:3. There are 3 different levels of omega saturation is there. If you remember that talks about that you have a single bond, and single bond gives you stability. 2 double bonds, 3 double bonds. So 18:1 is high oleic, linoleic, linolenic. डबल बॉन्ड बढ़ते जा रहे हैं, जितने डबल बॉन्ड बढ़ेंगे उतनी ऑयल की क्वालिटी खराब होगी. Okay, so if you have only single double bond, that is in oleic, is the best. Okay, so by transgenic manipulation, the oil quality has been modified because the pathway was stopped after oleic to linoleic or linoleic to linolenic. So there is a gene called fatty acid desaturase 2, FAT2, FAT2 naam hai isme, FAT2 gene that converts oleic to linoleic, linoleic acid, linoleic. So you can silence through RNAi, you can silence FAT2 and that stop the pathway there. So oleic will be accumulated. Okay, so the quality of oil will be, so better quality oil will come on your plate rather than high double bond quality. Or hai kuch, I think I'm almost done. Yeah. So whatever work I have shown you was done by Dhananjay Pandey. That was my PhD student. Now he is a faculty, and the work was supported by DBT, DST, CSIR, and this is my US collaborators, Jonathan Mendel and Josh and Ron. Those who are, Ron is in Israel these days, so we always work together to do some experiments. Thank you very much. If you have any questions, we can take it in the next 2 minutes. Otherwise, I assume. So I have talked about modifying your crops by saving losses, by increasing net yield, and by increasing nutritional quality. Overall, it is increasing the crop. So this is called crop improvement. विनोद जी, भाई आपके स्टूडेंट सब गायब हैं, कहाँ है ये? इधर ही है, 4-5 मिसिंग है 20 में से. अच्छा, चलो बैठिए. So we have talked about, and if you could do this one, you are basically reaching towards sustainability, right? Sustainable food for next generation. So these efforts, and there are many more efforts. So if you count these efforts in the direction of making your crop sustainable, crop resilient to climate change, you can do n number of experiments where heat stress, drought, salinity, other insects, fungi, bacteria, all these stresses can be addressed. Okay, so people are doing that in different crops, different stresses, and then you are basically modifying net production so that enough produce can be produced at last, and that can give you little security in your head that food is available. Okay, so I stop it here with these examples and with these narrations. So if you have any questions right now, I can take it, or you can contact me anytime. Later on, my email ID was there. Yeah, this is our sustainability. This is our workshop, and it says science today, sustainability tomorrow. So if you do something today, it will have a field impact after some time, isn't it? Thank you very much. Well, we should ask this question to government, right? Because I'm a developer. Because government, every country has a regulatory issue. Regulatory issues are regulation, I would say. Regulation is that whenever you make a product and that product is going to field, right? Is it safe? The first question comes, is it safe? And when you say safety, what is safety? Safety means it's my safety first, consumer, and then other species as well. For example, BT cotton. So BT cotton is having a gene, a protein ultimately, that can kill one insect, Helicoverpa. Question came that it is safe to environment. That means there are n number of insects in environment, including pollinators, and they are required. So is it that this gene can affect them also? Right? Well, the question. This is safety. This is biosafety question. That whether the modified product that is you are putting in the field, is it safe? Safe to whom? Safe to consumer first, and then safe to other species as well. If there is a butterfly that comes on cotton, on BT cotton, and eats a leaf of it, they do. So is Criminase gene going to affect butterfly also? Monsanto said no, it will not. That is a biosafety issue, right? So this is called regulation, because they have performed assays that the protein was fed to Helicoverpa, it died, fed to butterfly, fed to other insects, because honeybees, that means you should not affect other species, because pollinators are all simple in case of insect. So it was tested, and then the science was proposed that this protein that I am producing is very specific to Helicoverpa midgut. It is not binding to butterfly, and they have shown the data. That means it is, and assays also. This is called biosafety regulation issue. So regulation is very important. So whenever we, and because those assays were done by Monsanto in US, so we have taken it as such and we released it in. When we develop something, such kind of assay should be there. This is called regulatory concern of government. So whenever we develop, suppose we developed golden rice. So is that rice safe to everyone? Okay. Safe means there are 2 genes from bacteria, 1 gene from another plant. There are 3 genes came into it. So those regulatory issues come into picture when you have those biosafety experiments done. So if it is done with all products, and it is like you have to do those assays from n number of species, see if the pollen is going into air and affecting other crops, citrus crops or not. Those, that data has to be there. Additional data. What we have done so far, that we have developed the product in country, but those kind of safety data are missing largely. If it is there, you can always apply to government and request for it. So biosafety issues remain with plants. Biosafety issues remain a major concern before the regulatory approvals. Should be there, and there are there. It's not that they are not there, but that apprehensiveness. For example, BT cotton के बाद BT brinjal was developed. नाम भी सुना होगा आपने. And BT brinjal was never approved by government of India. Reason that people did not accept it. There is called a public opinion on any product. If government is releasing something, they always ask public. They put it on the website and ask that whether you would like to. Opinion is taken. It's not your decision is taken. Your opinion is taken, right? So the apprehension is that BT is a toxin from Bacillus bacteria, and it has a toxin, and that toxin kills one animal species, one insect species, Helicoverpa. It could be toxic to mammalian cells. Monsanto has clearly showed that it is not having any effect on mammalian cells. The data is available in patent, right? So it should be safe, but still we are apprehensive that it might affect mammalian cells. If we eat, so if we eat BT cotton, we don't eat. If I have this shirt from BT cotton, it's fine. But brinjal I am eating. So data is shown that it is not affecting. Now, if in your lunch, if I had 2 dishes made for you, a normal brinjal curry and a BT brinjal curry, who would like to eat it? Raise your hands. Who will eat it? Hello. 1 person. So out of 20, 1 person said, and I'm with you, two people said that I would eat it. Because I know the science behind it, that it won't affect. This is called apprehension. I'm not saying that you are wrong. It's your apprehension. Right? And if you are apprehensive to something, it's your choice. Right? So, the answer is right here. But you asked why it is not released, you would not eat it. Would you? So even if I am government and I am releasing it, would you buy it from the shop? Would you cook it? Would you eat it? Right? Public opinion is public acceptance is also. As I said, अगर हम खाएंगे बोना गेहूं खाएंगे तो हम भी बोने हो जाएंगे. Apprehension है, public opinion है. So until unless something is accepted by public, the consumer, it won't have a face in the market. Okay, even if I release it and my brinjal dies out on the shop itself, what would happen? Right? The public acceptance of any product. You go to market and buy any products. Let's say from you want to buy a soap, and that soap comes from Dettol. बड़ा famous है, Lifebuoy. So that brand has a value. Public acceptance. क्यों इतने advertisement आते हैं with Shahrukh Khan and Amitabh Bachchan? The public acceptance goes higher of the product. Okay. So if you have a product on the shop and you don't buy it, oh, it is not good. Product will die out. Right? There would not be any demand. If there is no demand, production would be hampered. It will die. Nirma sabun, Nirma soap. देखा है? एक वक्त था Nirma was 99% houses Nirma was used for cleaning the clothes. आज है कहाँ गया? विनोद जी को पता है. Nirma washing powder कहाँ गया? Public acceptance has gone down because it was harsh. कपड़े साफ तभी होंगे जब साबुन harsh होगा. It will clean. उससे हाथ भी खराब होने लगे. And public opinion said, no, this is bad. Another wheel came and then घड़ी came. They said, no, हाथ भी अच्छे रहेंगे. So it will be it is out. Public acceptance is another. Biosafety issue is one. Answer to your question after long discussion. Biosafety is one. Biosafety essays is one. Second, public acceptance. That is missing due to lack of education. You don't know how BT gene, criminase protein, kills helicobacter. You don't know, and but you know that it kills. And then if that protein is there in this dish, it may kill me too. If not kill, at least affect me adversely, right? It will not kill me. Fine, insect is small and big, but negative effect would be there on my health. If that is the case, you want to take it. But if you know the science behind it, I haven't shown it on the board, but if you know the science behind it, you would take it. So public acceptance comes with the knowledge. So knowledge, we have lack of knowledge. We don't, I mean, marine students don't know about BT cotton and vice versa. Maybe you don't know the botany what they are studying. Lack of knowledge, lack of other domain, and that leads to apprehensions. And if apprehensions are there, it's your world. Decide. Questions? Decide. Marine people.
Sir, just told the part where the only transgenic plant we use here in India.
Yeah, so far.
तो हम इतना एरिया क्यों लेते हैं? कंपैरेटिवली
हम इतना एरिया क्यों लेते हैं? हमारे पास उतना एरिया है. So we grow to maximum. So that maximum production is there. Yes, because production is less.
उनका कम एरिया में ज़्यादा प्रोडक्शन कैसे होगा? सेम बीटी कॉटन और हमारा
सेम बीटी कॉटन नहीं है. It's not about BT cotton data. Don't confuse. BT cotton is separate. Whatever data I have shown is for normal cotton. BT came into picture later on. So this is normal cotton data that comes, and ऐसा कम क्यों है? उसके लिए there are many factors as I mentioned. The germplasm is different. China varieties are different from. They are short, ours are tall, right? And they have more flowers. We have less flowers, less fruits. I would say. Biotic and abiotic stresses. In China, you don't have insects at all. Very few, because it is colder in region. So insects friendly nahi hai. India is like very beautiful climate for insects. Soil is different. Fungal and bacterial contaminations are different. Virus is not there. We have a lot of virus. So different factors: soil quality, biotic, abiotic, germplasm, they influence. And of course. And the challenge is that we cannot bring those varieties here in India to grow them because they are adapted to that region. That is a problem. So we have to modify our own varieties. Those are adapted here and make them compatible to system. So and then when BT cotton came into picture, so not everyone is growing BT cotton these days. Okay? We still have large area under normal cotton, and that is affected by insects and other stress. So BT cotton is helping, but that is only a fraction of it. Okay, tea time. जो चाय पिलाओ. चलो, thank you.